How the Cell Cycle is Controlled by Internal and External Mechanisms by Michaela Drake. The cell cycle is a complex set of stages from start to finish. It has 6 phases, each of which are highly elaborate. First is prophase, then prometaphase, metaphase, anaphase, telophase, and cytokinesis, thus making up mitosis. The first step is prophase. In this step, the chromosomes condense and become visible. Then spindle fibers emerge from the centrosomes. Next, the nuclear envelope breaks down the centrosomes. The centrosomes move toward opposing poles. This sets up for the second phase, prometaphase, in which the chromosomes continue to condense and the kinetochores appear at the centrosomes. The mitotic spindle microtubules attach to the kinetochores, thus setting up for phase 3, which is metaphase. During metaphase, chromosomes are lined up at the metaphase plate, and each sister chromatid is then attached to a spindle fiber that originates from opposite poles. Next, anaphase occurs. During anaphase, the centromeres split in two, and the sister chromatids are pulled towards opposite poles. Then certain spindle fibers begin to elongate the cell, thus preparing the cell for telophase, which is step 5. The chromosomes arrive at opposite ends and start to decondense. Then the nuclear envelope material surrounds each set of chromosomes. Next, the mitotic spindle breaks down. The spindle fibers push the poles apart. The last step is cytokinesis, which occurs differently in both plants and animals. In animals, a cleavage furrow separates the daughter cells, but in plant cells, a cell wall separates the daughter cells. And this is the end of the cell cycle. During cell cycle checkpoints, a cell passes G1 checkpoint as it enters the S phase of the cell cycle. During the S phase, what occurs is DNA replication. Then, after DNA replication occurs, the cell leaves the S phase and enters the G2 checkpoint. The G2 checkpoint. This is when the cell prepares for mitosis and meiosis. As you can see by the diagram, that as each checkpoint is fulfilled, there is more cell growth and DNA synthesis and formation of daughter cells as the cell cycle occurs through all the 6 phases that were discussed earlier. The checkpoints are covered in order to make sure that everything is going smoothly and there's no mutations in the DNA before it's turned into daughter cells at the end. Internal and external signals provide stop-and-go signs at each cell cycle checkpoint. The most important checkpoint is the G1 checkpoint because regardless of if cell division occurs, this process or checkpoint is still present. These checkpoints are important in order for a cell to regulate the cell cycle because it ensures that damaged DNA is not passed on to the daughter cells, which is important when preventing disease, specifically cancer. As you can see in the diagrams, there is unreplicated DNA and then a stop sign. The stop sign represents where there's a checkpoint in order to make sure that everything is going properly within the cells. And if not, apoptosis can occur, removing damaged DNA or damaged so that it's not replicated. So there will, in order to avoid mutations. Cyclins and cyclin-dependent kinases control the cell cycle. CDKs are a group of protein kinases that regulate the cell cycle. CDKs are also heavily involved in regulating transcription of DNA, mRNA processing, etc. The CDK's purpose is to bind a regulatory protein that is known as cyclin. The purpose of cyclins are to control the cell cycle by activating enzymes. As you can see, the cyclins, it shows in the diagram how the cyclins bind to the kinases, it's just shown in the diagram. Cancer results from disruptions in cell cycle control. Cancer is caused by the disruption in cell cycle control. Apoptosis occurs when the cell senses that the DNA is damaged. If apoptosis does not work, the cells with damaged DNA are able to divide, thus leading to the accumulation of further mutations, which then has the possibility to cause further complications, such as diseases, more specifically cancer. This is in part the reason for certain tumors forming. The diagram shows that in normal cells, the positive factors are duplicated or cell divides, but if there are negative growth factors, there's no cell division. But this is opposite from that of a cancer cell, because in a cancer cell, whether there are positive or negative growth factors, the cells still divide, which is why mutations can occur and further problems, further health issues can result from these complications. So yeah, I hope this video was helpful in learning about the cell cycle and why it's regulated. Thank you.